Hello. Did my topic scare most people off? Yeah, kind of thought so. I know it's Sunday and not that late, so I'll try and not put you all to sleep. I know it's a bit of a daunting topic, but we'll try and get through it with some ease. My name is Darryl Hudson. I've done my PhD at the University of Guelph in molecular biology and genetics. I've been there the past 7 years or so, and having recently left, I've started this consulting company so that I can continue work on this genome project. Essentially, we're trying to sequence all the cannabis genomes that we've been getting our hands on, so that's kind of what I'm going to talk about today. To just give you a brief overview of what I'm going to talk about, we're going to go through a bit of the history of agriculture here. So mankind has obviously been selectively breeding crops for about 10,000 years, and cannabis is essentially one of the oldest crops that we've been working on. So it's quite interesting from that aspect. Agriculture, maybe you can see kind of the background here is a view from space that's actually showing all these agricultural farms, and it's caused the single largest transformation of the Earth's ecosystem. But it's essential to support the economy that we have. We like to think we're maybe an oil-based economy or something like that, but without the freedom from having to worry about where your next meal is coming from, we don't have any of this, really. So when you see these signs or bumper stickers that say farmers feed cities, it really is the truth. Without overproduction and agriculture that we have, we wouldn't be able to have all this extra stuff and be able to focus on the things that we do in our regular lives. On top of that, there's substantial agricultural increases that are projected to be required in order to feed the population. It is growing, and as a result, a lot of the stocks have started to deplete. So it's a major concern for both farmers and the economies of the world. Another concern is that today over 80% of mankind's diet is provided by the seeds from a few species, and typically these are grown in monoculture, which has resulted in a lot of increases in yields, but also has provided the susceptibility to disease and things like that. So keeping diversity is something that we need to focus on, as well as creating the best monoculture crops. What's interesting is cannabis, being one of the oldest cultivated crops by man, is not one of these 12 species that we currently utilize in our common diets. And it still has amazing nutritional qualities, and it's a multi-purpose crop that we should probably be incorporating into this regime. Again, it's multiple uses: food, oil, fiber, and medicine. And it originated in the Ukraine-Kazakhstan region, but was essentially spread by mankind across the globe for agricultural purposes. Just to give you an idea of what mankind has actually been able to accomplish with plant breeding, here's corn. You can see the ancestor of corn is this teosinte variety, which essentially produces very small seeds that are of very little nutritional value, or at least not a lot of them anyways. With modern corn, they decreased all the branching and increased the kernel size and ear size significantly so that it's a viable agricultural crop. Rice has essentially had the same thing happen to it, where the conventional ancient rices were tall and had very small seeds. They produced some mutant varieties that were much shorter and able to use higher levels of nitrogen, and now the modern varieties essentially have very few strong tillers that produce a lot of seed. Cannabis is kind of the same. Breeders have been very successful, especially in the hemp world, at creating diversity. You can see kind of on the left there a really tall fiber strain, and on the right a very short seed oil strain being grown next to each other. And this essentially comes from the 3 different varieties of cannabis that we have: the sativa, indica, and ruderalis. So just touching briefly on cannabis as a medicine, this won't be news to most people here, but I just want to cover it because it gets into what the details of kind of what I'm going to talk about. Is that patients want a consistent, reliable medicine. This is what Dr. Horton was talking about with standardization. Right? Standardization is key, especially for Health Canada to be able to accept this as a legitimate medicine. It has to be consistent and reliable. On top of that, patients want variety and selection. We know that one size does not fit all. One strain does not treat everything. So we're going to have to develop specific strains for specific illnesses. In order to meet the supply in Canada, obviously we need quality growers and suppliers of this medicine. And again, I think we need specific genetics. So breeders, cloners, all this stuff has to become standardized in this country for it to be acceptable as a medicine. to me, it's kind of like if you went to the pharmacy and walked in and said, I have some illness, and they just gave you a handful of pills and said, one of these might work, right? It's kind of like that when Health Canada just provides you a bag of ground up whole bunch of plants, right? I mean, they don't know exactly what's in there, and they don't know exactly how it's going to work. And I mean, you can just see the difference between a nicely grown organic strain and what Health Canada was pushing on people. So a bit of an overview of modern plant genomics. The first plant genome sequence was done in 2000, and so this was actually done at the same time, essentially, as the human genome sequence, both of which took about 10 years to accomplish. Nowadays, especially in the past few years, we've had vast improvements in sequencing technology, and so this significantly reduces both the time and the economic cost of completing genome sequencing. In 2001, we published the cannabis genome sequence. So this was actually, they created an online browser called the Cannabis Genome Browser, which I'm going to do a brief demo of in a few minutes. But this is really a major step forward in understanding what's actually inside the cannabis plant that makes it medicine. So the idea now in 2012 and 2013 is to get our hands on as many varieties of hemp, both high THC cannabis strains as well as fiber and seed oil strains, ruderalis strains, to try and sequence all of them and be able to analyze the specific diversity in all these strains. So here's the actual paper that was published, and some of the initial findings that they were able to determine out of this is that the primary difference between the THC production is the expression of this one specific THCA synthase gene. So that's pretty interesting. And it definitely provides a framework for future studies. So since then, there's already been a number of publications that have come out of this work using the genome data to analyze specific proteins and what they do. So what's next? Most of the time when we look at all these pictures, we're looking at the phenotype, and so that's essentially how a plant looks. The phenotype really is an expression of both the environmental conditions that a plant is grown in, as well as the genotype of that plant. Breeders have been really good at selecting phenotype. We're getting really good at determining what environmental conditions are necessary to grow the best cannabis. The genotype information is completely what's lacking, and that's where we come in. That's what we're trying to provide. So this allows you to annotate the genes, essentially determine what they are and what they do. We often do this by correlating it to the phenotype and proving the gene function experimentally by either knocking this gene out or changing the expression of it so that we can actually see what expressing this gene does to the plant. Also, because there's so many genome sequences available now in plants, we're able to compare this to other species and determine the differences between species and why cannabis is the way it is. But we can also compare within cannabis and see why specific strains have specific properties that they do. So really, this will allow a number of things. It will allow the selection of specific genes by breeders. It will provide increased speed of selection, and it will also enable the use of modern biotechnology, which they've been using in corn and rice and other agricultural crop breeding for a number of years now. So the idea is kind of to build this big phylogenetic tree. So here we've got all the different species of plants that have been sequenced, and you can see originally they used to take the phenotypic properties, so how a plant looks, and make these trees, and they've had to rehash them all since genome technology has come about, because now we compare the actual genetic code and are able to get a much better understanding of how individual species evolved. Our idea is essentially to do this within the cannabis family so that we can determine exactly where all of these fall and how they're related to each other. Arabidopsis is the model organism. It was essentially the first genome that they sequenced. And like cannabis, it was found all over the globe, and you see a number of different phenotypes essentially coming out of this. So they've been able to do this with this plant, sequence all these different genomes and determine the exact genes that are essentially specific to the phenotypes that you see. Getting to the Cannabis Genome Browser, so this is available online. You can just Google Cannabis Genome Browser. The entire genome sequence is available for public download. It's about 90,000 Microsoft Word pages if you want to look at 90,000 pages of A, C, T's, and G's. Have fun with that. But if you are a scientist, you can actually start to use that information and go into things like the NCBI BLAST program. This is where you'd actually paste these sequences in and determine what they are. So I'm just going to do a quick little demo here and show you a bit of that. So here's what it looked like when you actually go to the website. Right here where it says download here, you can get the entire sequence, 90,000 or so pages. But really what this starts to show you is that we've been able to take individual scaffold sequences here and correlate them with a name and a function and start to determine exactly where these enzymes lie in a biochemical pathway. And so already they've got the precursor pathways as well as the actual production of THC and CBD quite well mapped out. And again, this is only published in 2011, so they're making progress quite quickly in this sort of thing. So you can also see the hemp finola variety there, and just to confuse the hell out of you for a second, this is actually what the information for a geneticist would look like. It's showing all the different scaffold reads here. Yeah, pretty boring. But one thing you'll notice is we've already started to add new strains in here. So originally we had the purple kush and the finola, and now they're starting to add in this USO 31, which is a seed oil variety, as well as the Chem Dog. So our hope is really to expand this and put hundreds and hundreds of these strains in here so we can actually see what's different between them. So just some interesting studies. I found it kind of curious that Dr. Hornby was talking about apoptosis. Originally they took the THC gene and linked it to this expression gene called GFP, which just glows green inside the plant. And what you can see when THC is linked to GFP, it completely goes out into the glandular trichomes. It's actually not located inside plant cells at all. It's totally spit out into that glandular trichome, which obviously we've known for a long time the trichomes are where the majority of the THC is held. But what's really interesting is when they tried to express this inside protoplast cells, which are essentially just plant cells floating in a media, it actually killed the cells. So THC is toxic to cells, and what they've found is at high concentration, it's toxic to insect cells, to fungus cells, to bacterial cells, and to plant cells, which is quite interesting, but also explains why it's excreted at high levels to the outside of the plant and not kept within the plant. It's essentially a very good defense mechanism molecule. It's going to keep away all your pests, but it attracts mammals, which are going to disseminate your seeds at the same time. So it's a very good molecule to have for a plant on an evolutionary scale. So here comes the fun stuff, getting into transgenic cannabis. Obviously, this is the inevitable next step. Whether we like it or not, people are going to make transgenic cannabis. I know pharmaceutical companies are already working on this. It is going to happen. The question is, when and who is going to do this? So yeah, transgenic means we're going to actually, rather than use a traditional plant breeding, you're going to specifically move genes around. And so you can see that kind of in the diagram. This green chunk of gene you're going to take out of a genome, you're going to stick it in this bacterial plasmid vector, use the vector to infect some Cali cells, so essentially some cells growing on a media, and from there you can actually repopulate a plant, which I'll show in 1 second. So unlike the traditional plant breeding where we mix two genomes together and hope we get what we want, this actually allows the specific selection and movement of individual genes in a controlled fashion, which is an interesting thing. I'll get into some of the possible uses of this in a few minutes. But so far, Cali Regeneration, so this is essentially one of the major steps in this process, is being able to propagate plants from a few number of cells that have been infected with this bacterium. This has been accomplished, and they're getting very well at it, as I'll show in a minute. Genes of interest, as I mentioned, are now being identified, so genes that might potentially be useful to us, we're starting to find now that we have the genome sequence. Transformation and regeneration has not been reported in the scientific literature. However, if you look online, you can find that it seems some obscure grad students have actually done this, maybe without their professors knowing. But there's definitely some images online of transgenic cannabis glowing different colors. So I don't know whether they're real or not, or just some Photoshop, but it's kind of interesting that they're close. And I actually expect within the next year that you'll see scientific literature on transgenic cannabis. And it has many, many applications. I'll get into a few just in a second here. So regeneration for Cali, essentially what you do is cut a little chunk of tissue, put it on one media, and transfer it to another type of media, and this is going to induce shoots to form. You can then transfer it to another media, which has a different hormone combination, and this is going to make roots to form. Originally in cannabis, this was very difficult to do. Now they now have some synthetic sort of hormone elicitor molecules, such as this TDZ, which is a cytokinin mimic. And you can see the Cali percentage up in the 90% now. So that's extremely good. So obviously there's the debate here. Is GMO technology good or bad? To me, it's a technology. GM is a tool. It's not good or bad either way. It's the use and the purpose which determines whether it's good or bad. The example I like to use is nuclear. When Einstein split the first atom, he didn't think that they're going to drop a bomb as the first application. But now we have power plants which are pretty safe and determined, to be a relatively good use of the technology. You can get even more simple though and think of like your common knife. People don't think of a knife as good or evil. It's how you use it. If you cut vegetables, the knife is not bad. But if you kill someone with it, it's probably not a good thing. So to me, GM is exactly the same thing. It's a tool that can be used either way. So far, there's definitely been an increased rate of GM use in agriculture, and this is because it provides benefits to the farmers. Farmers aren't scientists. If they grow something and it provides an economic benefit to them that doesn't hurt their families and is healthy, they're going to keep using it, and that's really what we're seeing happening. Unfortunately, there's been a lot of regulations involved in the GM process, a lot of backlash against it, which is understandable because essentially a lot of big corporations have used it to sell more pesticides and chemicals. Still, to date, there's no evidence that the GM technology itself actually has any adverse health effects. Essentially, a gene is a gene, and how it was put in that plant should be irrelevant. The fact of the matter is that we need to evaluate them all individually. If you're putting an insect gene inside of a plant, it's probably not the same as moving an endogenous gene that was already in that plant around and changing its expression level. It's completely different and should be evaluated differently. Lumping all GM in one category is essentially been a big problem for the GM crops that are going to help people because of the cost of getting through the regulatory process now. Only large corporations have benefited so far from GM laws. The only GM crops that are certified in North America are pesticide and herbicide GM. So at the same time, we've got vitamin A and golden fortified rice, which could be in Africa saving hundreds of thousands of lives a year, but is not certified because of the $500 or so million that it's going to require to get it through the testing and efficacy procedures. As a result, it's now been associated with one of these large companies because that was the only way for the scientists to actually get through that process, was to align themselves with a big corporation. Really, it's hurting the little guy, and we're seeing the same sort of thing in all sorts of industries. So, so it's an interesting question. My opinion is, again, we need to evaluate them all individually as to their merit and intent. One application for GM technology in cannabis, which I think is very viable and probably has no negative characteristics, is the autoflowering trait. So you see all these autoflowering strains, which are nice because they're small and produce fast, and so for a lot of growers with limited space or time, this is really an important trait. In plants, you have 4 conserved flowering pathways: the day length, vernalization, or cold temperature, gibberellin, which is a plant hormone, or the autonomous. Essentially, most plants will flower after a certain amount of time anyways. The Cannabisruderalis strain, though, grows at a really high northern latitude and has a short season, so this is what causes it to induce the autoflowering trait. However, it is not a very potent strain, and so when you cross it to a strong THC strain, typically the F1 is reduced by half or less than half. And so it requires a number of multiple generations of backcrossing and selection to get these autoflowering traits with a high THC content, and that's kind of a problem for breeders. With modern biotechnology, you could identify this single gene and move it around in a very controlled and precise fashion. You could create autoflowering strains in high THC strains without reducing the potency at all. And also, there's lots of precise control mechanisms where we can use inducible systems or lots of things where you could actually spray a chemical or a hormone on the plant and tell it to flower exactly now. So there's many benefits to this. Further applications are simple mutations. A lot of the agricultural crops are just mutants in a single gene, which cause beneficial phenotypic traits. THC hemp-free varieties for farming. Right now, farmers in Canada have to test their fields repeatedly, and if a single THC plant is found in there, they're usually in some big trouble. And so this is a significant cost to the farmer, and has been kind of detrimental in a lot of people taking this up in the agriculture community. As we were talking, standardization and tailor-made strains. I think this is really the future. And it's kind of a two-sided coin, where people were talking, do we want genetic diversity or do we want genetic specificity? I think the diversity is essential for creating the specificity, but from Health Canada's perspective, they want a standardized medicine. And some people, again, may want a high CBD strain with no THC, and we can really tailor-make these strains, start to create them in a fashion that's very specific to the disease. And I think the whole issue of the red pill, blue pill is a really strong issue, especially when it comes to a pharmaceutical in Health Canada. They want something standard, and if you can go in and say, I want this strain for my cancer, I want this strain for my pain, I want this strain for my epilepsy, that's going to be a significant step forward to being able to legitimize this as a standard medicine. And I think it's not only the way that things will go, but probably the way they have to go. There's also terpenes. These are some interesting molecules. It's typically the aromatic scent molecules in cannabis, which have been shown to have some properties, and you can see companies like Greenhouse Seeds starting to list the terpene content as well. There's been some reports, not necessarily scientific, but some things that I've read on the internet about autistic children eating high terpene strains, and they find that the THC difference isn't what's causing them to respond. It's actually probably these aromatic terpene molecules. So I'm not necessarily recommending we feed high THC strains to children, but it may be interesting to knock the THC out and use replacements, different strains for treatments, to replace drugs like Ritalin or all these things that seem to be pushed on kids a lot these days. And really what they've done in agriculture is primarily focus on stress tolerance, cold, drought, and insect resistance. These things we can definitely also provide to the cannabis plant. And they're getting really good at controlling expression. So we have tissue-specific expression. You can actually go in and transgenically change the expression of genes, but keep that in the leaves, or say only in the flowers, only in the roots, or have inducible systems where, like I was mentioning, you take something like ethanol or estrogen and you give your plant a mist, and all of a sudden that gene will be kicked in and activated like a light switch, and you can really control how things are happening inside the plant. So this is really some of the amazing uses that modern biotechnology could provide. I'm happy to take any questions. I just want to thank the collaborators, the University of Toronto, Saskatchewan, the NRC, Plant Biotech Institute, Nova Scotia, and I was at the University of Guelph. So you can really see this kind of a cross-Canada collaboration, essentially, yeah, across Canada, and we're hoping that we'll do really well with this in the next year. I want to thank Marco and the Treating Yourself staff for inviting me to come, and Greenhouse Seeds, who gave us a nice leg up by providing their entire collection for sequencing right away. So yeah, please contact me for any further information on this crazy stuff. Thank you very much. Happy to take questions.
All right, do we have any questions for Dr. Hudson?
What's your opinion on the future of, I guess easy way is privatization? Pfizer gets their hands on the cancer DNA genetics, and they get the patent on it, so they're the exclusive right to cancer, cannabis, wherever they decide to name it.
I'm completely against Big Pharma. So the whole idea behind this project is to make all this genome information publicly available so that the breeders can do what they want with it. We know Big Pharma has sequenced cannabis already. We know they are honing in on genes. But we've already kind of gave them a big kick in the ass by putting all these genes out there, because they're publicly available now. It's going to be very difficult for them to patent things that are already out there. And when it comes to genetics, you're not allowed to patent a gene. You're allowed to patent a process. So the process of them modifying that gene or using it for biochemistry, they can patent. But again, by providing this information to the breeders, we feel that the breeders will take it and run with it. They'll do what they do best, and they'll breed the best cannabis strains by being able to, rather than select for phenotype, select for the individual genes that are responsible for that phenotype. And it's going to add a lot of power to what they do. I think, yeah, big pharma, big tobacco, they're both interested in this. We know that. Are they sequencing it and making it public? Obviously not. They're keeping that for proprietary reasons.
No, it's once it gets into it, you're going to be number 2 after obviously.
Sorry?
You might be the next on the list because you're basically showing off how to get these GMA crosses done for yourself, if you will. But obviously these pharmaceuticals that are trying to get patent GMA for themselves and be able to market them as specific strains to their companies will not want an open source database. So I'm just thinking outside the box that might try to shut you down. So the future breeders can't really have access to that database once the DNAs are basically split across and people actually can modify their own or whatnot. You're probably already there. I'm just saying, you've already been doing it.
Yeah, you're going to see transgenic cannabis, and I think you're going to see it out of major corporations. My fear is that only big pharma will come out with a very few select specific strains, and they'll certify it through Health Canada, and you'll be stuck with what they give you, and that'll be it. I don't think that that's the way. I think when you look at big pharma, when you go in to buy Tylenol or aspirin, there's your generic brand, your store brand, your certified brand. I think that's the way it should be. You need to walk into a pharmacy and have selection, not be told this is the only thing that you can have. And I think the only way to avoid that is to get all the information public and get it out as soon as possible.